Because of photography, I ventured into the world of mycology because of the interest of the diversity of different fungi that I find in the woods and just about anywhere if one looks carefully. And once I started taking pictures of them, I had to figure out how to identify them so that I knew what I had and how to organize them, which is always a challenge for me to get everything organized well. Um, so with that, I just have built up a pretty good collection of images, several thousand of different fungi, obviously many repeats of the same species. But when you're trying to assure that you have a good reference collection of images to look at, it takes quite a few images to be sure you get all the details that are necessary for field identification. And then, of course, occasionally you have to go back to the office or the lab and do some additional work to truly get a positive ID. Some of the objectives for today is to review some of our layman terms that we use to describe the macrofungi that you will run into in different field guides of how to identify the fungi. Common names versus scientific names. I will attempt to give you the scientific names. I have them all. However, pronunciation can be a challenge at times, and Latin is not a living language, but it is what we use for our scientific names. We'll survey some of the diversity, and I had to selectively pick a few to include in the presentation to keep within the time frame, and hopefully I'll do that. And I will look at some of the different edibles versus the poisonous ones and try to cover that as we're going through. And I'm going to back up here a second to the title slide just to highlight the couple of images that I have here. On the left-hand side here where the pointer is currently, there are two different species in this particular image, one being the honey mushroom, and that is Armillaria malea, and the aborted entoloma down in front of them, and that is the Entoloma arbortivum. And the two of them interact with one another to generate this unusual looking structure in the front. The entoloma, when it's not interacting with a honey mushroom, does look like a standard mushroom. It's a slate gray color and has a pink spore production when it's producing its spores. Over here on the right-hand side, that particular sample is one of the bird's nest fungi. And the bird's nest fungi can sometimes also be called a splash cup because of the method by which their spore packets down in the nest are dispersed to spread the fungus to new locations for their food collection that they do. These are non-edible. There's really nothing there to be consumed. The honey mushroom and the aborted entoloma are both considered edible, but they can have some off flavors to them depending on who's consuming them. Now, starting into looking at some of the diversity of the fungi, what causes them or what kind of diversity there is. And the first thing that we see is the different shapes of that fungi come in. Of course, the first thing that we we'll look at is this standard mushroom. And you see here, this is one called the smooth paracel mushroom. And it's one that grows out in grassy areas. It's just a plain white mushroom, but it does give us all the standard details of the parts of the mushroom with the cap or pilus, under which the gills are presented. Gills can also be called lamellae. As we go down the stalk or stem, which is also called the stape, we'll see this ring around the stalk that is called the annulus. And the origin of that we'll see here in a second, all the way down to the soil surface where this particular fungus lives in the soil, not attached to anything in particular, where it acquires the nutrition it needs for growth and development. So We're starting out with the standard cap and stem type of macrofungi here. And most of the time we think of those that have gills. Looking at some of the other anatomy here, where that annulus or ring comes from, this particular mushroom possesses what is called a partial veil. And that veil is the covering over top of the gills when the mushroom is first growing, to protect them from abrasion as the mushroom pushes up through the soil. And when you go back and forth, this one would be referred to as a button mushroom versus the fully mature mushroom here on the other image. And you'll notice that the ring is already there. And as that cap begins to grow and flatten out and expand, the edge of the rim of that cap tears away from the body of that annulus ring or that partial veil, leaving behind the ring. And most of our mushrooms will have that partial veil, but depending on the species of mushroom we are looking at, some of them will be torn to shreds and disappear, or they may just be remnants hanging on the edge of the cap. Some of them will be very, very thin and web-like. So when they tear away and collapse down onto the stalk, there won't really be a whole lot of tissue there to be seen. And it doesn't show up until the spores start falling off of the gills, and some of them will collect on that remnants of that partial veil. As we look at the cap, here I've broken it open to show the positioning of the gills underneath the cap. Some of them are free from the stalk. Some of them will be connected directly to the stalk, and some of them will even extend down the stalk. And in that case, they're called decurrent gills. And then when we look at the base of this particular mushroom, there is no additional anatomy down here. When we get into looking at one of the Amanita species, the Amanita has a full veil as well as a partial veil. And in that case, the entire mushroom button is covered by the full veil. And as that button grows and tears open that full veil, it leaves a cup or a vulva at the base of the stalk, which is a very significant characteristic to be aware of whenever you're trying to field identify the different fungi. This one has no vulva. It did not have a full veil, and so there is no cup or remnants of a cup at the base of the stalk. And that reminds me to emphasize, whenever one is attempting to collect fungi from the field, you should collect the stalk all the way down into the soil so that you know exactly what is the base of that mushroom. If you just cut it off at the soil line, you could be asking for some pretty serious ailment. If not, death is potential. Again, this is a smooth paracel. It is considered to be an edible, but it varies in terms of the taste as to one really likes it or not. The next group are the cap and stem mushrooms with pores instead of gills. So initially, when you look at them, they don't look that much different than the ones that do have gills. It's not until you pick it and look on the underside that you will see the difference between the two. And yes, I know there is another species here in the image, but we're looking at the one that has the red cap on top. And when you look on the underside, you'll see that there is no gills present there. And when you look at it in greater magnification, you'll see that there are pores covering the pore-producing surface area. And this is one of the boletes. And this is a major characteristic of the boletes, is that they, instead of having gills, they have tubes, and the open ends of those tubes produce a pore surface. This one is the bicolored bolete, and it has a bright yellow pore spore producing surface, a red cap. The base of the stem is red, and then it transitions into yellow as you get closer to the cap. This one, if you press your thumb on that spore pore surface, it will bruise and turn a cobalt blue. Very fascinating color change on this particular bolete. And then when you cut through the cap and through the stem, it will slowly turn blue as well. Now there's another bolete, which is referred to as the bluing bolete, that as soon as you cut through that cap and stem, it instantly turns a deep, deep cobalt blue. That's not one you should be eating. That one is toxic. Now, a lot of the boletes are edible, but there are definitely a few that you need to know that aren't. You go from the gills to the pores to those that have neither gills nor pores and are referred to as the false gilled mushrooms. And this is one of the chanterelles, and specifically the smooth chanterelle. It is one that grows from the soil. And when you look on the underside of the cap area, there is no remnants of gills nor pores underneath there. It is just a smooth surface. And from that surface, there are tiny, tiny areas from which the pores are produced. The other one, the yellow chanterelle, which we'll see a little bit later, instead of being very smooth as the surface is here, there will be false gills or rolls in the surface of that tissue underneath. And then others that don't have gills include the very highly cherished morels. This is the yellow morel, and I really like the genus species of this one, Morchella esculatoides, and it kind of gives you that, when you get that Morchella, it sounds like something you should eat. Versus the false morel, this is the conifer false morel, and notice that all around it here you can see pine needles from the pine plantation where I discovered this one. Happened to be in Michigan rather than in Ohio. It looks similar to the true morel, but it is toxic. Now, some say they can eat these false morels without any ill effect. However, that is an individual reaction. There will be cases where they can eat them in small quantities, but they'll hit a time where that small quantity will no longer be tolerable and have some pretty major gastrointestinal response to eating the false morel. So not one that I would recommend venturing into that area too much. So knowing the difference between a false morel and a true morel is important. One thing that you can do is a true morel, when you cut it, bisect it and look to the interior, it is totally hollow in the center, including in the stem. Whereas these false morels, when you cut through them, there will be chambers, there'll be extra tissue on the inside, and it will not be totally hollow. So that's one of the field characteristics to look for to distinguish between the real morel and the false morel. And I will confess here immediately, all the images in this presentation are my own that I've captured, except for the morel picture here. The morels have still eluded me in terms of finding them at the right place at the right time. Now, this one happens to be from my niece, who was visiting a site that I was at only hours before and missed the presence of the true morel at that site. Then we get into more of the atypical caps, which the morel and the false morel would be an atypical cap, such as on the devil's stinkhorn that you see in this image. One of the really big differences here is it does have a cap. There is almost a veil-like cap over the tip of the stinkhorn, but it presents its spores in a very viscous, mucousy, sugary solution on the surface of the cap instead of on the underneath of the cap. And unfortunately, It will be non-edible. Well, actually, it's edible, but to get it past your nose could be quite a challenge because of the stench that it gives off. And the purpose of that stench, we'll see here in a second. So that's the devil stinkhorn. This is the common stinkhorn. As you notice, this one I found in the middle of a cornfield. In fact, this entire cornfield was polluted with the stinkhorns underneath the canopy. Here, the common stinkhorn I found in a park in a tree ring. So it was growing underneath of a tree. And then finally, this one has the name of being the elegant stinkhorn, or another name is the dog phallus stinkhorn, because of its general appearance could be mistaken for a buried dog in the yard. However, the purpose of the smell is to do exactly what you see here in this image, and that is to draw in carrion feeding types of insects. And when they discover the stinkhorn, they will consume the sugary solution. They'll walk all through this, and when they fly off to a new location, they will disperse the spores for the different types of stinkhorns. So kind of an interesting relationship between insects and fungi. So I do occasionally get entomology in here. And then we go from the stem and cap types of mushrooms to those that are either called shells or brackets or conks. And a very typical example of those would be the artist conk, which is a Ganoderma type of fungus, Ganoderma applanatum. It is a perennial fungus. In other words, it will add on to its structure year after year after year while it is decomposing the wood that it is connected to. It, as you can see here in the image, produces literally millions upon millions of spores through the undersurface of the conch or shelf type of fungus here. Now, these can get quite large. You can get multitudes of them on a single tree. If the tree is still standing and alive, it would be considered a parasite causing a wood rot in the living tree. If it's a down dead log, then it's simply considered to be a decomposer. But it is a significant decomposer of different species of deciduous wood. One that isn't perennial is an annual species is called the resinous polypore. It's a very attractive polypore, especially in its very fresh condition. Notice there's a lot of fallen leaves here, so it does typically show up late in the season, late summer into the fall months, and it being an annual polypore, it is much softer in tissue than the perennial woody type of polypore that the artist conch is. It is fairly common. I can guarantee you I will find it every fall. It's called resinous polypore for when it is fresh and just beginning to grow. There will be droplets of liquid all over the surface of the polypore. And it'll have anywhere from a brownish coloration to almost a burgundy coloration in that liquid that is dripping off the surface of this particular polypore. It will pretty much decompose over the winter months, and there will be only little bits of remnants of it by the following spring. And then we won't see it again until the fall. Now, this is the underside of an artist conch, just to show you that it is a pored surface, and the pores on these are pretty small. So it almost takes a magnifying glass to be able to see the openings through which the spores are released out of this. And you can also see why it's called an artist conch, because it's a palette upon which things can be etched and sketched. And by doing that and letting it continue to dry out, They can be preserved for decades as a palette for drawing things. And I've seen examples of it that have lasted for quite a long time. Another polypore is the true turkey's tail. And the true turkey's tail, when you look on the underside, has very definitive, fairly easily seen pores on the spore producing surface. And the reason for pointing that out is because in a couple of slides here, we will see a false turkey tail. And the way to distinguish between the two is the pore surface. On the true turkey tail, you can see the pores. On the false turkey tail, you cannot. So that is definitely another method for field identification. Now, the turkey tail is a non-edible as well. There just isn't anything soft there to consume. They persist for a number of years, but they're only really active 1 year at a time. And so fresh specimens will be much more distinct and definitive in their color presentation. Another polypore, this one is dryad saddle or pheasant's back. It's another annual type of polypore, so the tissues of this remain relatively soft. Now, as they mature, they'll get tough, but they won't be woody. And they will persist through the winter, but by the next spring, they're looking pretty rough in terms of their appearance. It is considered edible, but typically you have to get them fairly young to be able to consume them very easily. The underside, the pores on this particular one are much larger and much easier to see with the naked eye. The coin is there for size comparison, so you can see how big those pores are in this particular one. Dryad saddle or pheasant's back can be produced in multiple layers, as you see in this one, or it could be a solitary specimen growing all by itself on a log or a stump here and there. This one is let me back up here. This one, timing for this one, you can find fresh dryad saddle from early spring to late fall. So it's a very persistent, very common species that can be found almost any time through the growing season. The next one is a much smaller polypore, and it is highly associated with deciduous tree branches, but even more so with shagbark hickory branches. It has this tannish brown color from above when it is fresh, but as it ages, it will bleach out to a white color. Not overly distinct when it is looked at from the upper side, but when you look at it from the lower side, its common name is actually hexagonal pored polypore. And sometimes it's also called the hickory stick polypore because of its affinity for hickory branches. And that geometric pattern on the underside of that little shelf is just absolutely amazing. And I've used this particular one as my screensaver for years, simply because of the beauty of that geometric pattern. And again, not exactly an edible type of fungus, simply because it's kind of tough and there's not much there to be consumed. Not all the shelf types of fungi have pores. There's a number of them that have gills instead. This one is a late fall type of polypore. Excuse me, it's not a polypore, it's a gilled shelf mushroom. And its common name is the orange mock oyster. And the orange mock oyster is not an edible mushroom. And so it's not one that should be pursued for consuming. It does grow on hardwood rotting logs. And you can see it does have almost a creamsicle orange color to it, especially when it is fresh. And it doesn't persist very long. So you're only going to find it during the fall months, and by the end of winter, they're going to be completely gone. It does have gills underneath. You can see some of the mycelium from which those shells are produced. It is a decomposer of wood, and depending on the orientation of that shelf, whether it's on the side of the log or whether it's on the underside of the log, it will have a different morphology to the appearance of the fungus. A very attractive one, nice for taking pictures, but again, it's not one of the edible ones. In even a little bit more stranger type of shelf fungus is this one called the northern toothed polypore. And initially, when you look at it from above, it's kind of not all that unique in its appearance until you pull it off and look on the underside, and you will see a collection of long teeth sticking down from the undersurface of that shelf fungus. It almost looks like a brush from the side, kind of an easily identified one because of those teeth that are sticking down, and thus the name, the northern toothed polypore. And it's from these teeth that the spores are produced in this specimen. It is an annual type of fungus, and so the tissues are relatively soft on that one, although it's not considered an edible fungus. So it's just more of a curiosity of nature than anything. Other toothed polypores include the violet-toothed polypore. It's a little shelf fungus. Again, from the upper surface, it's not all that unique in its appearance. In fact, it could be confused with a turkey tail, but when you look at it from the underside, you'll see that there are short teeth sticking down from there, and it's called the violet-toothed polypore because of the purplish coloration that you will get on fresh specimens. And this can actually be quite spectacular when you look at them from underneath. When they're overhead, you can see how purple they truly are. And it's a very, very common one. I find it in just about every woodlot that I go into. And it is persistent, but it's still somewhat of an annual fungus. It's tough, it's woody, it's non-edible. I mentioned earlier the false turkey tail, and this is false turkey tail here. And from above, you can see why it is sometimes confused with turkey tail. It has that same concentric ring pattern to it. It grows on dead wood. In fact, you can find turkey tail and false turkey tail growing on the same log. But when you look on the undersurface of these false turkey tails, there are no visible pores. Even with a magnifying glass, you're not going to see pores through which the spores are produced on this fungus. And so it's a very attractive fungus. It tends to be bigger in size than the true turkey tail, but it is easily confused with the true turkey tail. Other shapes that we get with the shelf type or conch shape types of fungi. This is another woody one. It is very persistent. It will remain on the trunk of the tree that it grows on for years. This one is fairly specific to oak trees. And from above, again, it doesn't seem to be all that unique in its appearance, but when you flip it over, you'll understand where it gets its names. This looks like, if you use your imagination, a maze, a hedge maze. And the common name of this one is the thick mazed oak polypore. So another type of gilled polypore, and it's not just straight gills, it's kind of a maze pattern that you see underneath. So when you find a polypore that looks like this on an old dead log, you can be fairly certain that the old log is an oak tree. Again, it's a very woody type of fungus, so it's not going to be an edible one. but it's truly a unique one in its appearance. Then we get into those that are neither cap and stem or shelf fungi, those that are presented as either round or ball shaped. And here we get into things like the pear-shaped puffball. Again, an incredibly common fungus that we run into in woodlots. It is a wood decomposer of dead logs, and it can be produced in magnitudes of individuals all on the same log. And sometimes they grow directly on the soil. This one is called the spiny puffball. Here you can see an immature version of it, and then you see a mature version where the spines on the outside, that layer breaks away to reveal the puffball beneath and the opening through which the spores will be ejected out of that little puffball. Some of the puffballs are much tougher in nature. This one is called the earth. Sorry about that. This one's called the earth ball, and its genus name is scleroderma, and it's a very, very tough coating on the outside. And when you cut this across and expose the spore producing area on the interior, it tends to be purple in color. This is toxic. This is not a puffball to be consumed. The other puffballs, such as the pear-shaped puffball, as well as the giant puffball, when they are completely white on the interior, they are edible. But as soon as that interior begins turning color, it is no longer an edible fungus. Here's another type of puffball. This one happens to be the purple spored puffball. And it's kind of halfway between a giant puffball and the pear-shaped puffball in terms of its size. They get to be about the size of a loaf of bread. And they're called purple spored puffballs because when they are mature and are ready to release their spores, and they do that by simply the surface breaking open, it will be truly a violet to purple color on the interior. Now, some of the other puffballs are not edible. There's one that looks very similar in shape and size to the purple spored puffball, but it's yellow in the interior. That one is also toxic. So you have to be very careful of which puffball you are collecting. The next shape that we run into are the cup shapes. Here is one that has the common name of common brown cup. Most of the other fungi that we've looked at to this point have been in the group called the Basidiomycota, except for the morels. These are in the Ascomycota, or the cup fungi. And there are some other anatomical differences between Basidiomycota and Ascomycota, but its spore-producing surface is on the interior of the cup. And they have a pretty interesting genus name. It is the Pizizia, and almost looks like pizza when you look at the spelling just on a first glance. And there are several different species in that particular genus. Now, one that's much smaller than this common brown cup fungus is the eyelash cup. And you can see why it's called the eyelash cup, and because when you get close enough and high enough magnification, you can see the edge of the cup has little hairs all around it that look like little eyelashes. In these cup fungi, the common brown is an edible fungus, but the eyelash cup is not. And it's probably more so because of its size. The biggest of these are only about the size of a dime, and most of them are much smaller than that. So it's more of a curiosity to look at than it is to consider for eating. Then we get into the coral-shaped and the club-shaped types of fungi. Here we have a sample of the crown-tipped coral fungus. And the crown-tip coral fungus is edible, but there are a number of other coral fungi that are not edible and in fact are toxic. So you have to be very, very certain of your identification when it comes to the coral fungi as to which are edible and which are not. Then in this image, the actual subject of the image were the Indian pipes. And it wasn't until after I started looking at the images on the computer that I noticed that I had also acquired images of a club fungi. And this one happens to be referred to as the Golden Fairy Club. It's pretty tiny, obviously, compared to the Indian pipe that's growing beside it. And so because it's so small, it takes so much effort for collecting, it's never really been determined as to whether it's edible or not. And then there's everything else. And there's a whole bunch of other shapes and sizes and colors out there that we can encounter in the woods, such as these yellow fan-shaped jelly fungi. They're pretty small, growing on rotting logs. We've already talked about the bird's nest fungi, which are too tough to be considered edible. And there's a couple of different species of those that we can encounter. And then there's those that are called cauliflower fungi. And it does literally look like a cauliflower growing on the forest floor. And the cauliflower fungi are edible. So there's a multitude of other shapes that once you start looking, is quite interesting. One thing that I often get asked is, how do I find them, or where do I find them? And the where is they can grow on soil anywhere in the woodlot, such as the russola that we have here. And this one is most likely the emetic russola. Many of the russola fungi are edible, but this one is not. As its name implies, the emetic. An emetic is something that makes you throw up. So this particular one you would not want to collect for eating purposes. Others grow on wood, such as the chicken of the woods that we have here. And this is one of the top 10 edible fungi that we can encounter in woodlots, and is a prized collectible for consuming. I like them simply because of their photographic quality. The colors are just spectacular. And there's actually probably a multitude of chicken of the wood fungi species because there's a ton of variation in their appearance. There's one that is distinctly a different species than this one. This one is sulphurous, that is called cincinnatus, which is much pinker in color on the top and has a pure white undersurface, where this one tends to be yellow on the undersurface. So there's a multitude of fungi that all collectively get called chicken of the woods. Then there's even fungi that grow on fungi. The little mushroom that's being chowed down upon by the slug there is called the bleeding mycena. And the reason it's called bleeding mycena is when you pull them off the wood that they're growing and snap the base off, it will literally generate a deep burgundy colored liquid out the base of the stem that will stain your skin and look like a blood stain on the skin. However, you'll notice that this one seems to be incredibly hairy. Well, that's because there's another fungus that is parasitizing the bleeding mycena, and it's called the bonnet mold. And bonnet mold will destroy the cap of the bleeding mycena. And the first time that I encountered that, it was just spectacular. The amount of growth that was coming off of these little bleeding mycena mushrooms in a woodlot. Now, another thing that produces a lot of diversity amongst the fungi is their size. And this is where it takes a keen eye to discover some of them. Here you see a hickory nut that I had picked up, and growing on the shell of the hickory nut was this tiny gilled mushroom. Now, I was not able to identify this one to species, partially because it may have never been studied and given a genus species name, but they can be incredibly tiny. So you do have to be a sharp eye to pick up on some of them. Here is one called a crep, C-R-E-P. It's a tiny little white mushroom that grows on wood as a wood decomposer. You can see it doesn't get a whole lot bigger than that. But then there are some monsters out there amongst the fungi as well, such as this horse mushroom, which is an agaricus species. And it is one of the edibles. You could make 10 meals out of just one of these types of fungi. And this one happened to be growing outside of one of the university buildings on the OSU Lima campus. And there was a whole line of them growing there. Here's a huge sample of one of the comb tooth fungi that was growing on an old down oak log. And I'll come back to this one a little bit later when it comes to being able to discover things in the woodlots. Where do you find them? Besides being down on the soil, you can find fungi in just about every habitat you go into if you look carefully enough. So they grow from the forest floor to growing on specific trees, to growing even up in the tops of the canopy. So if you notice right around here, there are some chicken of the woods growing at least 50 feet off the ground up in the canopy of this tree. These are chanterelles here, and this one is called the cracked cap Polypore. And the interesting thing about the cracked cap Polypore is that it is very specific to black locust tree. And so if you know that it's a black locust tree and you see one of these big polypores growing on the trunk, you know what it's called. Or if you know what the cracked cap polypore looks like, you can identify the tree species that it's growing on because of its close association and affinity for just black locust trees. And this one it was one of the samples that I found there at the Mansfield campus in Mansfield, Ohio. When do you find them? Yeah, almost all year round, especially when you're looking at some of the perennial fungi like the artist conk. You can find it any month of the year. But even some of the fleshy fungi, such as these velvet stem fungi, can be found very early or even very late in the season. So this is one of the very first fungi that begins growing in the springtime. And I can find them as early as late January, beginning of February, and as late as the end of December, with fresh specimens growing at that time of the year. Some of them are very, very common, so you can find them almost any time during the growing season. Some are very common, but they have an incredibly short growing season, such as the morels. We are probably in the beginnings of the heat of morel season, and it only lasts a week or so. Some are pretty rare, and you can only find them occasionally. And most are active between April and October. So here again, the velvet stem, you can find them throughout most of the year. And the oyster mushrooms can also be found in most of the year as well. For example, here's a fresh growth of oyster mushrooms on an old stump in a park that I stopped at over on the eastern side of Ohio. And the date that I took this picture was December 29th. And so these were fresh fungi almost at the end of the year. And when I got closer and was looking at them more closely, you can see that they even had water frozen on the surface of these fungi. So they will take advantage of just about any time of the year to grow, depending on the species of fungus that you're looking at. And then how do you find them? As Kathy mentioned there at the beginning, we do a mushroom class at the Mansfield campus most falls for the past couple of years, except for this last year. And walking out into the woodlot, I usually don't make it very far because of spotting different fungi growing on the soil, growing on the logs, while other people tend to walk right past them. So you kind of have to have a bit of a keen eye or a mindset to be looking for them. So how do I find them? Well, first off, I frequently visit the same woodlot over and over because at different times of the year, there will be different fungi growing. And I have at least 10 different woodlots that I frequent on a regular basis. 3 different college campuses. Ohio Northern University has a woodlot. OSU Lima campus has a woodlot, a rather large one. And the University of Bluffton has a nature preserve. And I visit all 3 of those woodlots on a regular basis. Metro parks. There's at least different 8 metro parks between where I live in Hardin County and where I work in Van Wert County that I visit on a regular basis just because there could be different fungi growing at different times. I walk the trails and I walk the trails in both directions because you may not see everything going in one direction. So you have to turn around and come back. As an example, you see this log here that has this brilliant orange chicken of the woods growing on it. When I walked the trail going one direction, I did not see it because it was up on a slope of the hill that I couldn't see it going one direction. But when I turned around and came back the opposite direction, it was just spectacular. And it was a very disappointing walk initially. One, because the mosquitoes were horrendous, and I thought I was just giving a blood meal to every mosquito in the woodlot. And then I wasn't seeing anything until I had given up and turned around and was starting back the other direction. I examined down logs, dead tree trunks, stumps, kick around in the fallen leaves to find different fungi, revisit sites a couple of days after a rainfall. It's not always a guarantee, but it does, in some cases, stimulate new reproductive growth of the fungi after a rain. And slow down. Just take your time and really look at the ground, look up the sides of trees, etc., and look for splashes of color. And again, that's how I find many specimens deep back in the woodlots is due to coloration. Here we have summer oyster mushrooms in the woodlots I'd say about 8 years ago. Because of all of the dead fallen ash trees, the oyster mushrooms were just incredibly numerous throughout the first couple of years after the ash trees had fallen to the ground. And here you can see one that's hidden underneath a log. This happens to be a bitter bolete that was growing underneath a log. And it probably was the little polypore that drew my attention to the log in the first place. And then I discovered the bolete growing underneath it afterwards. The other thing we have to remember is that the fungi change in appearance as they're growing. And so going back multiple times can eventually tell you what you're seeing out there. And here you can see some fungi growing out of the stump here. And on initial glance, it might be hard to identify what it is. Kind of just looks like a couple of rivets coming out the trunk of the tree. Now, coming back a little later, you start getting better definition of what the species is. And even later yet, it's more fully developed and finally reaches its full potential in size. And of course, that's the dryad saddle or pheasant's back. And in this particular case, all stages were present on one tree here. and didn't have to return frequently to see as it developed. Timing can be everything. This was a specimen that I discovered in one of my woodlots that I frequent. It was the first time I ever discovered this one, called the Berkeley's Polypore. It's a rather large polypore, very attractive. This one was absolutely a fresh specimen. And putting a coin on it to give you some size reference, it was pretty big, and I'd never seen it there before. Well, the next year I went back, nothing. The year after that, nothing. Year after that, and on, until finally 6 years later, it finally generated the reproductive structure again. If I hadn't repeatedly gone back to the same area time after time, I would have never seen this particular polypore again at that same site. And I'll back up here. It has a great affinity for oak trees, and unfortunately it is a parasite, and it does cause some pretty severe root rot and butt rot on oak trees. So it's not exactly one you want to see growing on your oak trees in your yard. Now, I'll take about another 30 minutes here and go quickly through a number of unique looking fungi in the woods, and starting out with this one here, which is a cap and stem type of fungus. Has a very unique appearance on the upper surface, and those are all kinds of hairs that are kind of bunched together. It looks like it just got up out of bed, so it's kind of a mess on top. When you look underneath, it has a pored surface, so it is a bolete. And a unique thing in this particular case, because it was over top of this log, all of its spores were accumulating on the surface of the log. So I got a spore print at the same time that I got the fresh specimen. And this one is called the Old Man of the Woods bolete. Those that collect fungi for consumption, you might be salivating at this point because of these huge lion mane toothed fungi. They're not very fresh, so they probably wouldn't be ideal for consuming. But you can see they are very large specimens of lion's mane on this very old log that has generated these pretty consistently for a couple of years in a row. And you notice that there's a stream here. This is in one of my frequented metro parks, where the metro park is actually on that side of the stream, and this was actually on private property on the opposite side of the stream. And I had to find my way through the stream to get to them to actually take good images of the lion's mane. And it's a pretty good stream to have to find a crossing point on, but it's a pretty consistent log for generating lion's mane. A relative of lion's mane is bear's head. Here's a degrading specimen, and here's a fresh specimen. Now, the interesting thing was that this was November 2nd that this was present on the tree trunk, and this one here was in September. So it's another fall producing type of fungus, another cherished edible when they're fresh and clean, and very unique in its appearance with those long teeth that it generates. And the third in that group is the comb tooth jelly or fungus, which has a much shorter tooth that it produces, and it more almost looks like stalagmites and stalactites sticking out of that one. That earlier comb tooth that I had shown, that was about the size of a bushel basket. That was another one that I discovered from a great distance off the trail, simply by keeping a watch for splashes of color in the background. And when I got closer to it, there were multiple specimens on the trunk of the tree. Some you can see had already completely degraded and disappeared, but this one was a fresh specimen. And again, this was November that this was still actively growing. Very unique appearance to it. And the interior, it almost looks like cauliflower once again, with the density of the teeth and the branches inside of that large comb tooth fungus. Unique colors. This one's called coral pink Merulius. It's a very fleshy, almost an odd feeling shelf fungus. Whenever I picked it, false gills on the underside of it. Partly that's where it falls in the Merulius group. But a very distinct color, and it can be deeper pink than this one, but a unique one. I find it often associated with downed American beech trees. So kind of a unique specimen. Another one that's kind of weird looking is the tree ear or the wood ear. And this one I found in August of the year, but it's a fairly persistent one. One of the unique characteristics is when it gets droughty and dry, these will dry out and just become very leathery, but next time that it rains, they will rehydrate and puff back out again. And you can see why it's called tree ear. It kind of looks like an old man's ear when you look at it at particular angles. There are incredibly toxic fungi out there that can be encountered. The first time that I found the destroying angel, which is one of the Amanita bisporidia species of fungi, it is distinctly one that has a full veil that leaves behind a cup or a vulva at the base of the stalk. Pure white, has an annulus ring, white gills, white cap, white stalk, and incredibly toxic. So it looks similar to that smooth parasol that we saw at the beginning of the presentation, but distinctly has that definitive cup at the base of the stalk. These can be hidden down in the soil, and that's part of the precaution of digging these specimens out completely before collecting anything for consumption. This one, called the Wrinkled Peach, is a very interesting type of fungi. It almost looks like a piece of candy there, a jelly candy that would be peach flavored, or you might think it'd be peach flavored. It is a stem and gilled cap type of fungus. This one's a little bit older. It's not an entirely fresh specimen. This one was much fresher than the previous one. So you can see more of that definitive peachy color to it, but it's the upper surface of the cap that is the most unique characteristic. It has this incredibly reticulated surface to the top of the fungus. It's said to be a common fungus, but one rarely finds them. I've been lucky enough to find multiple specimens over the years. Next, it's not the white one we're looking at here. It's this dark black one in the background called dead man's fingers, kind of an eerie type of fungus. And here you see a younger stage of it, a little bit older. It's getting more macabre looking with that gray, the black appearance. Sometimes they're bifurcated, but you can see where it gets its common name of the dead man's fingers based on the appearance of them. And if you find this in a base of a tree in a cemetery, you might think twice about what you're looking at. A related one is the Candlesnuff Fungus, very much skinnier, but it has a similar appearance to it. And as it ages, it does get grayer and blacker. But it's also frequently encountered on decomposing logs. This one, called the wine cap mushroom, it's a pretty big mushroom that grows directly out of the soil. But the unique characteristic about it, not only its color at the top of the cap, but it's the annulus ring underneath. It almost looks like a cogged wheel around the base of the stem in that annulus ring. Kind of a unique specimen, grows directly out of the soil, so it's not dependent on decomposing wood. Another one that can be found growing directly out of the soil are the milk cap mushrooms. And there's a ton of different milk cap mushrooms out there. Some of them are quite edible, some of them are not. And many times they are associated as mycorrhizal species with different types of trees. And unfortunately, they do grow amongst the poison ivy. Fortunately, I am fairly immune to poison ivy. And it has kind of a funnel-shaped cap when it's fully mature. But the unique characteristic is when you brush across the gills or crack the gills, they ooze a latex out. And that latex kind of looks like a milk. That's the common name, milk caps. And depending on the species that you encounter, it could be a yellowish milk, it could be a white milk, a pink milk. That's part of the characterization. Notice the gills are directly connected to the stem on this species. Then we get into, here's another confession. These are not my images. These are from Asia, because there's a group of conch-shaped polypores called the varnished or lacquered polypores. And the ones that are most relished in Asia are Ganoderma lucidum. Ganoderma lucidum was mistakenly used as a genus species name here in the United States. However, with some recent genetic studies, it has been determined that we have next to no Ganoderma lucidum here in the United States. There's only a couple isolated populations in California and Utah, and they probably are escapees from cultivation. In Asia, China, Japan, Korea, etc., Ganoderma lucidum is used extensively in traditional folk medicine. And that's why it's cultivated extensively there. They grind it up and they make tea out of it. And that tea is supposed to have all kinds of medicinal purposes to it. And we have similar looking Ganoderma fungi here in the United States. But it has been determined that almost none of them are Ganoderma lucidum. So anybody who's looking for folk remedies to go out there to collect that particular varnished polypore to make tea out of, you're not using the right species. For example, here is a distinctly varnished topped polypore. It has a stem on it, so you can see there's a stem connecting it to this decomposing wood. There's an example of it exposed, so you can see that stem. As it ages, it still has a shiny, varnished appearance to it, but it loses some of that amber and yellow color and turns mostly to a reddish color. This one happens to be Ganoderma curtisi. And that, no, I didn't name it. That just happens to be somebody else had named it with that name, Curtis. Probably a last name rather than a first name. This is a very common varnished polypore that we have in the US. The other one, you can see specimens, very large specimens around the trunk of this particular honey locust tree. Kind of almost looks like muffins growing out of the soil. It does distinctly have a varnished stem on these. And this is another example of Ganoderma curtisi. The other very common one that we have grows out of wood from stumps that have been cut off at the surface of the ground. And they can also grow on the trunks of trees. No stem associated with these. These are Ganoderma sessile. So sessile meaning they have no stem. And these two are very common. In fact, these are frequently encountered in turf grass in street tree areas where the trees have died and they've been ground down just below the surface of the soil. Here's another example of one that was off of the trunk of an old silver maple. And they do survive through the winter. However, it's an annual production. So this was last fall, this was this spring. And you can see that's the same polypore from one image to the other. Pretty much a dead polypore that will not regrow from that polypore. And you can see underneath, it was connected to the wood through this portion of that fungus. But also notice all the holes in the surface, undersurface of that. There were a huge number of pleasing fungus beetles that were hiding and overwintering underneath that polypore, and they do consume fungi as their regular meal. This one was another example of a stemmed varnished polypore, and it happened to be growing at the base of a pine tree. This was up in oak openings, and I just happened to go back to the same site the same year. This one was in July. This one was in August. So that's the same specimen, just a slightly different angle that it's appearing. And so you can see the discoloration. The other example around the other part of the tree from earlier in July till late in August. Since it's growing with a pine, this one is going to be Ganoderma sugi, or the hemlock varnished polypore. Very has a great affinity for conifers. And another couple here to go. We can have this one's called the little Blackfoot polypore. It tends to be earlier in the season when this one grows. It has a pored surface underneath, kind of a unique overlapping of the top of the polypore. And it's only about 2, maybe up to 4 inches in diameter, and grows on dead logs, not edible. One that is related to it is the big Blackfoot polypore. It's also called the bay-colored polypore. These can be huge. These can be up to 10 inches in diameter, also growing on down decomposing logs. Here, the one that I'm holding in my hand is just one of the smallest specimens that I pulled off the log. So that's it right there, just to show the underside doesn't quite have the same overlapping characteristic on the top of the polypore. And this one happened to be a wonderful example of an entire log lined with that big Blackfoot or bay-colored polypore. Spectacular specimens, non-edible, too tough to be able to be consumed. Here's another example of the same one that I ran onto in a local woodlot. You can see that it's growing at the end of a downed log. These were pretty fresh specimens. So I decided to go back a little bit later, notice the rolled edge of it here, just to make sure that it is the same specimen. But when I came back to it, it had a very different appearance to it. Now, the day that I took this image, I used flash because it was overcast. It was almost ready to rain. So I took a quick picture and then ran out of the woodlot, not realizing that I had captured a second organism growing over top of the polypore. And you can see it on the log here as well. And this is called plasmodial slime mold. And this is one that will send homeowners screaming out of the woods thinking that there are aliens or body snatchers growing in the woodlot, because you can see this incredibly neon green yellow coloration, and it almost looks like veins running across the surface of these polypores. Then under magnification, you can even see pulsation going through these branches of this plasmodial slime mold. And it literally moves like an amoeba across the surface of the log. And so this can migrate in a fairly short period of time from one end of the log to the other. And you can see it's stretching forward like an amoeba going down across this log. Now, really unique. Even though it's called a slime mold, it's not a true fungus. It's a protozoan type of organism. This one I found early in the spring on a grapevine, and it's called the orange grapevine slime. It's actually a combination of about 3 or 4 different fungi, including a yeast that grows on the sap that can come out of grapevines. Down south, it's called deer vomit, such a nice common name to use for it. But again, rather alien looking, growing on the grapevine. Here we have another unique example of a fungus that has an unusual relationship. This one happens to be a sooty mold. And this sooty mold has a very intriguing genus species name. And genus species name is Scorius spongiosum. It almost sounds like a Harry Potter casting spell with its genus species name. But it has a specific relationship with an insect. And the insect is a woolly aphid. Now, there on the Mansfield campus in Mansfield, Ohio, it is the beech blight woolly aphid that we find this fungus associated with. In this case, these images were taken in Sharon Woods in Columbus on black alder. So this is the alder woolly aphid that is in association with that sooty mold. And what's occurring is when the aphids feed, they release an excessive amount of liquid through their anus, and we call it honeydew. And this mold grows on that honeydew that accumulates underneath the colonies of the aphids. And it produces a really spongy growth, and it can be huge, and it can be persistent. It will stay there all winter long into the springtime, and it will turn black. So it almost looks like a gall growing on the surface of the branches of the trees, but it's a mold that is consuming the honeydew from the aphids. So again, there's the woolly aphids, sometimes called the boogie woogie aphids because they do a dance when they're disturbed. But there's the fresh and aging sooty mold that grows on that honeydew. And to end it out, I'll throw one last insect in there that has nothing to do with fungi. Here you can see a caterpillar called the harvester caterpillar. And you can see one over here as well on the other side of the branch that are associated with these woolly aphids, which are maple woolly aphids. And the bizarre thing is these caterpillars are predaceous. They actually eat aphids for their nutrition. So a carnivorous caterpillar. And I just happened to be there at the right time, at the right place, when the adult butterfly, the harvester butterfly, was laying its eggs in amongst the colony so that its caterpillars could parasitize or predate on the aphids growing on, this happened to be greenbrier plant that was being fed upon. So all kinds of weird stuff in the woods. So we'll bring that portion of the presentation to a halt, and we'll address questions at this point.
Okay, thanks, Curtis. Awesome, as always. And don't feel bad about the morels. I know that I have lots of them in my woods out back, but it takes me to find the first one to be able to find more. And there's years I never find.
You got to get that mind's eye set to be able to see them.
Right. And so I haven't found any yet this year. So
they should be up.
Yeah, they should. But I haven't seen any. I probably walked on a few, but.
Okay.
Okay, so let's take a look. We've got some questions. Let's see here. So the first one says, show aborted
entoloma.
Entoloma, please.
I think she wants to see an actual entoloma that isn't aborted. So give me a second here. Go on to the next question. I'll find one.
Question, fungi have no roots?
They're not plants, and so fungi do not have roots. fungi have mycelium, and the mycelium is a quote unquote pseudo root. And I didn't go into that kind of detail in terms of the anatomy of fungi, where they have two different types of mycelium. The one mycelium, which is a group of threads that is the body of the fungus, is a feeding mycelium that is in whatever substance is being consumed by the fungus. It digests outside of its body, absorbs the nutrients, and then that feeding mycelium will extend into the reproductive structure that we call the mushroom. And so when you pull out pick up a specimen of like a cap mushroom, you might see the threads coming out the base of the stalk. Now that is a mycelium that is a quote unquote pseudo root. It's not a true root. Okay. And so, no, there aren't roots.
Another question, what are the gills for?
The gills are the surface upon which the spores are generated. They're in the tissues of those gills are specialized cells inside of which the spores are generated by meiosis to produce haploid cells, and will have generated the reproductive structures for dispersal of the fungus to new locations.
Okay. So Neil asks whether DNR has one of their field guides for fungus or mushrooms, and I don't believe I've seen one.
I can't say that I have either.
Marnie might know of one if they have, but I haven't seen one. So whether it's in the works, because they have added a lot to their collection recently.
Are you talking about Division of Wildlife?
Yeah.
Yeah. Let me look. I have them all right here in front of me. And I think they have one on lichens, but I don't think they have one on mushrooms.
Okay.
Yeah, I did.
Yeah, they have Common Lichens of Ohio, and I don't.
That's right.
That's the one they have. Yeah.
Okay.
Now OSU has their publication, but it doesn't cover a ton of them, but it does cover a lot of the common ones. Mm-hmm. And so for the question about what does a non-aborted Entoloma look like, there's the non-aborted Entoloma mushroom there with the aborted version of it behind it. So you can see it has somewhat of a steely gray coloration to it. And I'll pop that down for a second. Oops. Stop sharing for some reason.
Now.
Oh, I know what I did. And I'll just put up another example to show the spores of that. Am I still sharing? Oh,
nope.
No, hold on. Where'd it go? I hate Zoom at times.
Mm-hmm.
Yeah, you're seeing a whole bunch of examples of them, but you can see the pinkish spores that the Entoloma produces. There's a pile of them there, and I don't know whether it'll allow me to do this. Nope, I see it, but you don't. Stop sharing a second. Try again. I know there's a better way to do this. And so there's that pinkish spore from the Entoloma mushroom. Kind of a unique color for spores.
Yeah. So Abby asks, what tips do you have for making spore prints for ID?
Oh, okay. Of course, you need to collect a relatively fresh specimen, but not too fresh, because if it's not ready to drop spores, then it's just going to degrade and not produce a spore print for you. But well, once you collect a specimen, I would use 2 different colors of paper, one a dark color, one a light color, because you can have white spores and you can have dark brown spores depending on the species that you're looking at. So you'll overlap the 2 and put the cap of your interested fungus on top of that division, and then put a bowl over top of the cap so it doesn't dry out too rapidly. And if it's really ready to give off spores, less than 24 hours, you'll have a spore print.
Okay.
And just lay it or position it somewhere where you're not going to have any kind of breezes blowing across it.
Okay, so the next one says, if you see a shelf-type mushroom on a living tree, will it kill it?
That's a loaded question.
It is a loaded question.
Well, first thing that we have to remember is that the shelf type of fungus on the outside of the tree indicates that, yes, there is a lot of more fungus on the inside of the tree. Now, how aggressive that fungus is at producing a decay depends on the species of fungus and also the species of tree. Some trees are very good at compartmentalizing decay on their interior, while others are not. So that's a hard question to give you an absolute definitive answer of how serious it is or isn't. If you have a shelf fungus growing on the trunk of a tree, usually it's not good news.
I usually tell folks if it's growing there, it's been growing inside the tree for a while.
Oh, could be decades that it was
Right.
growing on the inside of the tree
Before it shows its external. Okay, so what effect does temperature extremes have on the emergence time of morels?
Yeah, soil temperature probably moderates a lot of the swings in aerial temperature. So the air temperature may not be as significant as the soil temperature. And so we're probably looking at soil temperatures around 50 degrees, somewhere 45, 50 degrees Fahrenheit before we start seeing them emerge out of the soil. So the aerial temperature swings may not be as significant as the soil temperature.
And we need moisture, usually.
And it has been
Dry
really
spring.
dry.
Yeah, dry springs usually aren't as good for them as, it seems like you don't find them as much. But Abby asked, what's your favorite mushroom ID guide?
I have a stack of them, and I use them all. The most recent one, and I used this one earlier this morning. It's called The Mushrooms of the Upper Midwest. This is the 2nd edition. It's called A Simple Guide to Common Mushrooms, Mushrooms of the Upper Midwest. And this is brand new. The 2nd edition just came out, so it's very up to date, has some decent pictures in it, and is fairly easy to use. But that's just one of several that I use on a regular basis. Another one is Mushrooms of the Northeast, and I've worn this one pretty good. The cover's pretty flapped up because of thumbing through it frequently. The Audubon field guide is okay. But there could be a number of specimens in there that may not be common in the Midwest.
Right.
So yeah, any field guide I can get my hands on. There are definitely some that just get kind of put to the side. I just purchased my first copy of what's it called, Mushrooms Demystified. And for all the press that it gets, it wasn't as exciting as I thought it was going to be when I got it. The author is from California, and so most of it does concentrate on California.
Okay. Let's see here. Are dead man's fingers stinkhorns?
No. Nope.
Linda asks, I'm always hesitant to harvest fungi when I hike in the woods simply because I fear depleting them. Am I correct, or any guidelines?
Well, again, you have to remember that the objects that we see and that identify the mushrooms on are the reproductive structures. And they only represent less than 1 to 5% of the overall body of the fungus. And the fungus does not depend on those reproductive structures for its existence. They are primarily for spreading the mushroom to new locations. So collecting the mushroom, if you have your favorite morel spot, by removing the mushrooms, you're probably not going to deplete the fungus from that area. The thing that's going to result in the fungus disappearing is a lack of food.
Okay. How do you improve mushroom biodiversity in a forest or private property? Should people transport spores or decaying wood within county limits, of course?
I don't have a good answer to that one. Again, just like any other living organism, they have all kinds of environmental requirements for their existence, whether that is a type of food that they prefer, to moisture content, to pH of the soil, to temperature ranges. So there's all kinds of factors that would need to be taken into consideration. So simply taking the spores or the a reproductive structure from one wood lot to another may not guarantee that it can establish there. And the other thing is, you got to remember, these fungi are generating millions, if not billions, of spores from those reproductive structures, which blow on the wind. So if it's going to get there, it's probably already blown there at one time or another. So if it's not growing there, it's probably because there's something not being met in its environmental requirements.
Okay. Are there any fungi that are just toxic just to the touch?
No, that's a quick one. You have to ingest them.
I've heard you should soak edible mushrooms in salt water before cooking. Do you do this? I do that with my morels, but I've always done it. At least my mom always did it, and she said it was to, make the bugs expel out of the. Whether
That's
there's any truth.
Primarily, what you're doing is encouraging insect larvae that you don't necessarily see to leave the tissues of the fungus.
Great. Okay. About the honey mushroom, armillaria. At the base of a large silver maple cut down 3 years ago, armillaria mushrooms develop at the trunk's base and large structural roots. Problem: when the fungi release spores, they melt into a black runny mass that hosts other fungi and stinks. The stink lasts about 2 months, and during that time, we can't use our patio close by. If I remove the fresh mushrooms, can I prevent the smell?
Well, certainly by removing that biomass, there's not going to be anything there to break down and smell. And most likely what you're dealing with is the ringless honey mushroom, which was Armillaria tabaciens, which has actually been reclassified into Des Armillaria. So it's DES Armillaria.
Really?
Yeah, very recent reclassification.
Okay, that's good to know. Let's see. Here's one for you, Curtis. What information can fungi provide for use in timber operations and forest management?
Well, if you have a lot of fungi growing on the trunks of your trees, you're not going to have very good lumber.
No, it is. It's an indicator that there's decay going on. And so the tree itself probably isn't really solid,
right?
Not, at least not completely solid.
And the quality of the lumber could be diminished. Yeah. The other thing I've seen, and this occurs across the United States with Armillaria malea, which is the shoestring honey mushroom, it can wipe out a woodlot.
Yes.
It can decimate a woodlot.
Yeah. Yeah, I have seen Armillaria, the shoestring root rot, just take out a stand of white oaks in a relatively short span of time.
There at the OSU Lima campus, it took out all the sugar maples.
Right? Yeah.
Hopefully that answered your question.
Are there any non-native invasive fungi?
Probably. Now we're going to push the borders out away from just macrofungi to Dutch elm disease. That's a non-native invasive fungus that killed the American elm.
Okay.
I'm not sure about the chestnut blight, whether that was non-native.
That was non-native. Yeah.
So a lot of our disease-causing organisms are fungi.
So was the one with thousand canker.
Yep, with walnut.
That's also
well,
it's not native to Ohio.
Right. It's native to the western. Yeah.
Right. Let's see. Can you find fairy pins in more than violet-toothed polypores?
You got me on that question. I'm not sure.
Okay.
No,
okay.
Unfamiliar with the terminology.
Do we have any bioluminescent fungi in Ohio?
Absolutely.
We do.
Yeah, one of them is the jack-o'-lantern. In fact, I think it's difficult to see at times simply because it's got to be really dark. And in most of our urban areas, there's way too much light pollution.
Right.
But if you go down to Hocking Hills or one of the forests where there isn't a lot of artificial light, I think you're going to see quite a bit of luminescence in those types of scenarios.
How often do you use binoculars when you're out?
Not often. Most of the time I'm just looking at the ground. I just have to be careful not to run into trees when I'm looking down all the time.
That would be a problem, or stumbling over something that you should have been able to see.
And I guess I use my camera as binoculars.
That's
because
probably
it has a telephoto lens on it.
So Peter asks, what causes the bluing in some mushrooms when bruised?
Well, I'm not sure exactly what the chemical reaction is. It's going to be some sort of oxidation reaction because you're exposing the tissues to the oxygen in the atmosphere, and then it oxidizes whatever chemical is present there. And I don't know what the chemical is.
Okay, so Mark Wilthu says, please touch base again on false morel traits versus morels.
Well, one of the field identifiers for the morel versus false morel is the interior. A true morel is totally hollow on the interior when you bisect it from top to bottom, whereas the false morels have all kinds of extra tissues on the interior. So there's a lot more substance on the inside. Then the outside, when you look at the cap of the false morel, it's convoluted and kind of looks like the surface of a brain, whereas the true morel is pitted. And so you have ridges and deep pits in the surface of the cap.
Okay. Please name or identify the saddle fungus that had large pores on the bottom.
Dryad saddle.
Okay.
Dryad saddle or pheasant's back. And let's see, that is, where's my list of genus species? There it is. Cereoporus squamosus. And that's CERIOPORUS SQUAMOSUS. Dryad saddle, or otherwise known as pheasant's back.
Okay. Are you a member of the Ohio Mushroom Society, Curtis?
No, I'm not. I'm remiss. I should be.
Do you have a favorite fungi?
That's a tough one. It depends on the moment, I guess, as to what happens to be there for me to photograph. I guess the chicken of the woods tends to be, because of its brilliance in color, that tends to be a favorite just to see it. But even some of the tiny ones like the walnut mycena can be intriguing to see.
Are the seen parts of the mushroom considered the fruit?
Stretching the definition of fruiting body, yes.
Okay.
It's a reproductive structure.
Examples of fungi which would be found only around pine trees.
Oh, there is certainly a couple of different species of fungi that are mycorrhizal associations with pines, such as the chicken fat bolete, otherwise known as the slippery jack. Simulus americanus, I think, is the genus species name.
Okay. So let's see, you've answered some of these about specific fungi to particular tree species. Do you have any photos of your, what? Go ahead.
Well, some of there at Mansfield, when we went into the pine plantation, we had a lot of milky caps that were associated with the pines.
Right. Someone's asking, do you have any photos of spore prints on paper?
Oh, yeah.
To show.
Go ahead and ask another question. It'll take me a second to find it.
Let's see. Are there types of mushrooms that appear mostly after forest fires or prescribed burns? Boy, that's an interesting question.
Well, I don't know if it's specifically associated with a forest fire. However, there are definitely some fungi that only grow on burnt pine cones.
Okay. That's interesting. Are mushrooms important for wildlife?
Oh, lots of wildlife consume mushrooms. Anything from a multitude of different insects that specialize on mushrooms, to squirrels eat mushrooms, chipmunks eat mushrooms. You saw the slug eating the mushroom. So yeah, there are a lot of wildlife that consume mushrooms. Now, are they completely dependent on it? Not necessarily the mammals, but the insects that specialize on fungi, absolutely. Okay. So there's a spore print of the green spored paracel mushroom.
That's cool. Okay. Let's see. Are mica caps a good edible species, and are there some that should be avoided?
There's one called the alcohol inky cap, Caprinus. And the problem with the inky caps is when they get a little too old, they melt away.
Right.
And so if you don't get the inky caps early in their development, they're not very edible. So timing is everything in that one. But the alcohol inky cap, if you consume alcohol at the same time that you eat the inky cap, it will make you very sick. And in fact, a lot of the wild mushrooms, the recommendation is don't drink alcohol because it will activate the toxins even in the edible species of fungi. So it's not recommended to drink alcohol and eat wild mushrooms at the same time.
Okay. Are there fungi that are safe for humans and not for like cats, dogs, or farm animals?
It's mostly the dogs that we run into problems with them eating fungi when they shouldn't be, like the lawnmower's mushroom or the haymaker's mushroom, especially small dogs. If they consume them, they may have a very negative reaction to them. It's when you run into that, you get into that. The toxin or the poison is in the dose. And so a small-bodied animal wouldn't necessarily have to eat as many as a large-bodied animal for it to be sickening to them.
Okay. Differences of jack-o'-lantern and chicken of the woods.
Well, the jack-o'-lantern mushroom is actually a cap and stem gilled mushroom, whereas chicken of the woods is a polypore. Let's see, and I've got lots of pictures of jack-o'-lantern. It's actually an amazing specimen to see. So go ahead to the next one and I'll.
Can you tell what kind of mushroom it is just by the spore print?
Not necessarily. And, well, let me add to that answer is it is highly recommended that one always do a spore print in the process of identification. But when you really, really get into separating out species and verifying absolute identification, most of the professional mycologists will look at the shape and size and dimensions of the spores to be able to identify the fungus appropriately under a microscope, because they are microscopic.
Okay. Is the mulch vomit fungus a true fungus?
No, it's a slime mold.
And is there good treatment to get rid of it?
No. And sorry for the short clipped answer, but the slime mold that is the dog vomit is a decomposer of mulch. It loves hardwood mulch. And so as long as you use hardwood mulch, you have the potential of getting that dog vomit slime mold. There is no chemicals to treat to kill it off. And about the only way that you can reduce its presence is constantly stir the mulch, because the longer the mulch sits stationary, the more supportive of it is of the slime mold. And if you do get the slime mold, before it goes into spore production, lift it with a pitchfork and dispose of it before it gets a chance to release additional spores. So what's pictured currently there is jack-o'-lantern. And let's see if it'll let me advance through here. It's an orange fungus through and through. So its flesh is orange. Its cap is orange. It has a white spore, but so does chicken of the woods. You can see it is gilled, so you can see gills on the undersurface of the cap, and it grows much very similarly to the honey mushroom in terms of it'll grow a lot of caps from a common point. So you can see quite a few specimens there. There's a good side shot. So it is, the jack-o'-lantern is not a shelf fungus. It is a cap and stem gilled fungus.
Okay. Let's see here. Do fungi release spores all the time?
No. And so it's kind of bizarre at times if you're at the right place at the right time. I've watched spores being released from artist conch, and it looks like smoke coming out of the artist conch. So number one, it has to be mature enough to have fully matured spores, and then right environmental conditions under which to release them.
So there's a question about if you ingest the spores into your respiratory system.
Not a good idea. And so not that the fungus is going to germinate and start growing in your lungs. However, it is a foreign body. It can be irritating to lung tissue. It can cause asthmatic responses in individuals. It can cause inflammation in the lungs. So no, it's not highly advisable to be sucking up spores. In fact, if you listen to the news forecasts carefully, part of their pollen count is pollen and spore count. And so they're counting mushroom spores floating in the air to advise those who have asthma and other respiratory ailments of the potential for irritation.
So here's a good one for you. What caused your interest in becoming a fungi expert?
I'm an insect expert that has taken up mycology as a hobby. And the interest was photography, digital photography. They were subjects to be taken pictures of.
So what resource do you recommend for insect-specific parasitic fungi?
Insects that eat fungi or fungi that eat insects?
Doesn't specify that. I think it's the relationship between insects and specific fungi.
I don't know. I don't think there is a resource that concentrates just on that kind of a relationship. So it's a matter of just attempting to look it up on the internet.
Yeah, and there's, every different guide has a little bit different twist to it. It's kind of like when I'm like you, except with tree books. I have stacks of tree books, and some of them are great for one thing, and some of them are better at others. And so, you accumulate a collection that has some.
And I can't say that I've, even in all of the field guides for the fungi, I don't know that any of them have been even made reference to the insects that feed on fungi. And there's a ton of different beetles that feed on fungi. And there's a bunch of different flies that feed on fungi. But I can't say that I've ever seen a guide that specializes on that particular subject matter.
Are there any rules of thumb for distinguishing edible from non-edible?
No.
The ultimate question that we get.
The main answer to that is learn to identify. Learn to how you identify the individual species, and find, like the Ohio Mushroom Association, join a group like that. Find somebody amongst that group that is incredibly well versed in identifying, to be able to learn yourself. Don't just go willy-nilly out there and start collecting fungi and eating it. There's that old adage of there are old mushroom hunters and there are bold mushroom hunters, but there are no old, bold mushroom hunters.
Are mushrooms parasitic, decomposers, and symbiotic?
Yes. They cover the gamut. They cover the whole spectrum. About a third of the fungi are decomposers. About a third of the fungi are symbiotic, mycorrhizal, and about a third of the fungi are parasitic. At least the ones that we know. There's a whole bunch of them out there that nobody has done a thing with.
So there is the question about picking mushrooms. Is it legal in Ohio? And I answered one previously about, harvesting on private property. You do need to have permission.
Yeah.
when you're on private property.
And when you're in state or national or even metro parks, it may be completely prohibited to do so.
As a woodland owner, I want you to ask first and not have me find you down in the woods with a bag full of mushrooms. So I will speak from a landowner's perspective. If you ask, you're probably going to get permission. If you don't ask,
You're going to get run off.
yeah.
And just like any, you just got to use common sense when it comes to collecting in woodlots. You wouldn't go just traipsing in to hunt with a rifle for a deer. Well, you shouldn't.
You shouldn't. As a woodland owner, I can say that that does happen sometimes, but yeah. Let's ask, we'll do one more and then I'll post the link for the survey. When harvesting edible mushrooms, is there any benefit to leaving the base intact with the soil/wood?
No. There's just nothing there that is of significance once the reproductive structure is done.
Okay. So I'm going to post the link in the chat box. It just went up. Hopefully everybody can see it. Fill out name, first name, last name, and either ISA number or SAF number. And then I will submit those to the national office for both entities for you to get credit for today's awesome seminar on mushrooms. If you've never